Hello everyone, I'm Dr. Ashish Guragai, and today we'll be talking about innate versus adaptive immunity, and then we'll be talking about B lymphocytes and T lymphocytes. We'll be focusing on their development and their activation. So let's get this started. So let's start with innate versus adaptive immunity. So what is innate immunity? Innate immunity is nothing but our body's defense mechanism against all the pathogens that we encounter from minute to minute in our daily life. So, why do you think we don't get upper respiratory tract infection all the time? Or why do you think we don't get skin infections all the time? It's because we have this innate immunity components such as physical barriers, for example, skin and mucosa. And we have ciliary motility and cough reflexes, which prevents us from getting infected by inhaled pathogens. So when we inhale a pathogen, ciliary motility and the sputum traps the bacteria, and then ciliary action expels the sputum, which has the trapped bacteria inside them. Similarly, the cough reflexes also help us to remove these pathogens from invading our respiratory tract. Now, the cellular components of innate immunity are macrophages, neutrophils, dendritic cells, natural killer cells, and complement proteins. Similarly, we have secretions like our saliva, which contains lysozymes. Similarly, we have gastric acid secretions in our stomach, which protects our body from being invaded by the pathogens that we consume. Now, the components of adaptive immunity are T cells, B cells, and the circulating immunoglobulins. So let's talk about the response to pathogens in innate immunity. This response is non-specific, which means our components of the innate immunity, they cannot distinguish whether this is bacteria, whether this is virus, or they cannot distinguish whether this is gram-positive bacteria or gram-negative bacteria. All they do is they recognize that these proteins or these antigens in these pathogens are foreign and they are trying to invade our body. That's all innate immunity does, all right? They know these proteins or antigens are foreign and we have to destroy them. That's all they do. All right. And the response they create is non-specific. Now, another important thing about innate immunity is that these reactions occur rapidly. All right. So let's say you get a cut in your hand. All right. Inflammation occurs within minutes. Similarly, because of the disruption of the physical barrier, that is skin, now the bacteria can invade our skin. But do we get an infection after every cut? No, right? We don't get an infection after every cut. Why does that happen? Because neutrophils and macrophages will rush to the site of inflammation and any bacteria which invaded our skin will be phagocytosed. All right, and this happens within minutes to hours. Now, talking about memory response in innate immunity, there's no memory response. That means our innate immunity does not remember whether Neisseria caused infection in our body or there was a streptococcal sore throat. All right. Our body does not remember the pathogens. All right. Now, let's talk about adaptive immunity. The components of adaptive immunity are T cells, B cells, and the circulating immunoglobulins. Now, compared to innate immunity, the response to pathogens in adaptive immunity is highly specific. For example, if there is a streptococcal sore throat, there will be a specific response to that particular strain of streptococcus. All right, it will be highly specific, and that response won't be same to another bacteria. It will be completely different and unique. The immunoglobulins that were produced against one strain of streptococcus won't be same compared to another strain of streptococcus. So that's why adaptive immunity is highly specific. But the thing is, this specific immunity develops over long periods of time. It does not happen rapidly. And during that duration, it's innate immunity which protects us from the pathogens. All right. During the development of adaptive immunity, we need some kind of protection, and that protection is provided by innate immunity. Now, unlike innate immunity, the memory response in adaptive immunity is vigorous and quicker, right? In innate immunity, there's no memory response, but in adaptive immunity, the memory response is really robust. Now, what does that mean? It means if we get infection from the same streptococcus after, let's say, 2 months, our response will be really quick and we don't need a long period of time to adapt. The response will be robust and there'll be specific immunoglobulins to destroy that particular strain of streptococcus. Now let's talk about pathogen recognition. So we talked about how macrophages and neutrophils go to the site of inflammation, but how do they recognize bacteria or pathogens? How do they recognize, okay, these are not our normal cellular components of our body, they are foreign. So how do they recognize it? Now, this happens from toll-like receptors. So toll-like receptors are a pattern recognition receptors. That means they recognize different proteins or different molecules in bacteria via these toll-like receptors. Now, let me give you a specific example. So, toll-like receptors can recognize pathogen-associated molecular patterns or PAMPs. All right. Talking about gram-negative bacteria, they have lipopolysaccharide. And our toll-like receptor recognizes lipopolysaccharide and knows that this is a foreign pathogen, this is a bacteria, and the body knows that we need to destroy these pathogens. Similarly, there are other PAMPs like flagellin, nucleic acids, right? And our body is able to recognize these proteins via the toll-like receptors. Now, in adaptive immunity, this pathogen recognition occurs via immunoglobulins. Each immunoglobulin has a unique binding site to a particular antigen, and when that antigen is exposed, our immunoglobulins go and bind to that antigen and then trigger adaptive immunity. Similarly, like immunoglobulins, there are cytokines which are able to recognize the pathogen and initiate the adaptive immunity. Now let's talk about functions of natural killer cells. Now you might be wondering why I am only focusing on natural killer cells which are part of innate immunity, but there are other cells as well, right? But the reason I'm focusing on NK cells or the natural killer cells is because from the exam point of view, these are super high yield, all right? So what are natural killer cells? They're nothing but a type of lymphocytes which secretes porphyrins and granzymes to induce apoptosis of virally infected cells or tumor cells. Now, this image shows how natural killer cells identifies a pathogen and then releases porphyrins and granzymes. Now, if you look at this image, you can see that there are immunoglobulins which are binding to the antigens, right? Now, this will form an antigen-antibody complex, and our natural killer cells in the body are able to recognize this antigen-antibody complex, and then it will know that it needs to destroy the cell, right? This way, natural killer cells release porphyrins and granzymes and cause apoptosis. Besides this, natural killer cells are also able to recognize pathogens via PAMPs, right? Now, the activity of natural killer cells are enhanced by interleukin 2, interleukin 12, interferon alpha, and interferon beta. Now, these are super high yield information. Now, from the exam point of view, how they will ask a question about this activity enhanced by these interleukins is that they'll give you a question of viral infection. They'll explain how a kid got virally infected. And then they will say that natural killer cells were able to destroy this virus infected cell. Which of the following interleukins enhance the activity of natural killer cells? So this is how they ask question, and there'll be different options, and you have to choose one among these four. The high yield ones are interleukin 2, interleukin 12, interferon alpha, and interferon beta. So these enhance the activity of natural killer cells. All right, and these are super high yield from exam point of view. Now, there's one more really high yield information. These cells are also able to destroy any cell which lacks inhibitory signals such as MHC1 on the cell surface. Now, these are really important in tumor cells. Tumor cells, if they lack MHC1, it will signal natural killer cells that these cells do not have inhibitory signals. So these are definitely some kind of abnormal cells and natural killer cells need to destroy them. All right. Now, these inhibitory signals are really important because these signals protect our normal cells from being destroyed by natural killer cells. All right. Similarly, natural killer cells play a huge role in type 2 hypersensitivity by destroying cells via antibody dependent cell mediated cytotoxicity. We'll be talking about these in detail in our hypersensitivity video. As it is really important, I want to highlight this destruction of tumor cells by natural killer cell one more time. So the tumor cells, they lack MHC1, all right? And MHC1 is an inhibitory signal. If this inhibitory signal is absent, then it tells natural killer cell that this cell is some kind of abnormal cell and it needs to be destroyed, all right? Super high yield, that's why I repeated it multiple times. I want you to really remember this function of natural killer cells. Now let's talk about major functions of B and T cells. So these are just a basic overview of the function of B and T cells. So B cells, they initiate adaptive immune response via humoral mechanism. All right, the humoral immune response of the adaptive immune system is initiated by B cells. Similarly, they recognize and present antigen as well. The most important function of B cell is to produce antibody and also to maintain an immunologic memory. Similarly, the functions of T cells are to initiate cell-mediated immunity. There are different types of T cells. The most important ones are CD4 T cells, which helps the B cell to make antibodies and also produce cytokines. Similarly, there are CD8 positive T cells, which directly kill virus-infected and tumor cells. Now, T cells play a huge role via cell-mediated immunity to cause delayed hypersensitivity, which is also called cell-mediated hypersensitivity. It is a type 4 hypersensitivity reaction. Now, T cells also play a huge role in acute and chronic cellular organ rejection. So this was just a brief overview of function of B and T cells, and we'll be talking about them in great detail as the video goes on. But this was just an overview for you guys. Now, before we talk more detail about function of B and T cells, we need to understand HLA gene complex and MHC proteins. So our body has these gene complexes called HLA, human leukocyte antigen. So these gene complexes, they code for MHC proteins, major histocompatibility complex. All right. Now, MHC proteins, they play a vital role in initiating immune response as they present antigen fragments to T cells and bind to T cell receptors. Now, let's look at this figure. This is MHC class 1. It has alpha 1, alpha 2, alpha 3, and a beta 2 microglobulin. All right. They are also called HLA, HLA-B, and HLA-C. All right. And this is the cleft where antigens bind. Now let's look at the structure of MHC class 2. They also have similar antigen binding cleft, but their components are different. Unlike MHC class 1, MHC class 2 has two beta components, beta 1 and beta 2, and they have two alpha components, alpha 1 and alpha 2. All right, they're also called HLA-DR, HLA-DQ, and HLA-DP. Now let's talk about MHC class 1 and MHC class 2 in detail. So what is the loci that produces MHC1? It's HLA-A, HLA-B, and HLA-C. All right. And did you notice this mnemonic? One letter. If you notice, all the HLAs which produce MHC1 or which codes for MHC1, they contain only one letter, HLA-A, HLA-B, and HLA-C. Now, if you look at the loci that codes for MHC2, they contain two letters, HLA-DP, HLA-DQ, and HLA-DR. Now, this is really high yield mnemonic, all right? The USMLE exam loves to ask you about these components. So you really need to know this. Now, let's look at the structure. Like I said earlier, MHC1 has a heavy chain and a beta 2 microglobulin, and this heavy chain is formed by alpha 1, 2, and alpha 3. All right? Now, compared to the structure of MHC1, MHC2 has two equal length chains two alpha chains, alpha 1 and alpha 2, and two beta chains, beta 1 and beta 2. Let's look at the structure one more time. So this is the long chain or the heavy chain, which contains alpha 1, alpha 2, and alpha 3, and it contains a beta 2 microglobulin. Now, MHC class 2, it has two components, alpha and 2 beta. All right, alpha 1, alpha 2, beta 1, and beta 2. So this is MHC class 2, and this is MHC class 1.